Hello, hello. Hello, hello, hello. A very graceful morning to everybody who have gathered here today in the third day of 5th Annual Conference, Mcccon 2024 on Functional Food as Dietary Intervention for Chronic Diseases, organized by Medinapore City College. Today is also a very precious day for us, and it is the foundation day of our college. Medinapore City College has successfully and gloriously put its feet in the 8th year of its voyage towards academic enlightenment. It's a moment of revelry and festivity for us. I am requesting everybody to give a huge round of applause to honor this day. A huge round of applause, everybody.
A very good morning to everyone present here. On this auspicious day, we will have another Young Scientist Award session. We have with us today Dr. Shantanu Kor Mohapatra.
Big round of applause.
This session will be chaired by Professor Nirmal Kumar Sarkar. I would like to request Professor Nirmal Kumar Sarkar to come to the stage and to chair the session. Sir, over to you.
Good morning to everybody, and warm wishes on the foundation day of the college. We shall have a very brief technical session now. It will be a Young Scientist Award program, and in this session only one lecture will be presented, and it will be presented by Dr. Shantanu Kor Mohapatra. I welcome Dr. Shantanu Kor Mohapatra onto the stage. Let me introduce to you all Dr. Kor Mohapatra. This is only a formal introduction, because he is already known to everybody in this college. Dr. Shantanu Kor Mohapatra is an associate professor cum head of the Department of Paramedical and Allied Health Sciences in this college. He received his PhD degree from Vidyasagar University and subsequently worked as research associate at Bose Institute, Kolkata, and then worked as assistant professor at Shastra Deemed University. In the past 10 years of teaching and independent research career, Dr. Shantanu Kor Mohapatra has been engaged with the screening, optimization and intensification of macrophage repolarizing and therapeutics from different sources, synergic effects of drugs, immunomodulators, and nanomedicine in the treatment of visceral leishmaniasis, tuberculosis, and leukemia. Though quite young by age, Dr. Kor Mohapatra has shown commendable brilliance in the arena of his research. He has already completed or running as many as 6 research funds. Very recently, I think in the last year, Dr. Kaur Mahapatra as the principal investigator, along with Dr. Shravani Pradhan of this college, received a mammoth grant from ICMR amounting to rupees 88 lakhs for detection and management of tuberculosis in the tribal communities in the entire Midnapore belt. Thereafter, they received another ICMR grant amounting 50 lakhs of rupees for their work on drug delivery system, liposome-based drug delivery system. And finally, Dr. Kaur Mahapatra singularly received another SERB core grant of rupees 48 lakhs for isolation of anti-leishmanial agents from sugarcane waste. Akhir chibre ke kaje lagiye, he is trying for finding out some anti-leishmanial compound. He has already authored an international patent, and he has as many as 65 research publications in internationally peer-reviewed journals to his credit. Already 4 researchers have earned their PhD degree under his supervision, and another 4 are pursuing their PhD program under the supervision of Dr. Kaur Mahapatra. Dr. Kaur Mahapatra is also serving as a guest editor in Frontiers Journal, and he is also acting as the reviewer of several peer-reviewed journals. And quite importantly, Dr. Kaur Mahapatra has already been the recipient of owner of some prestigious awards. For instance, Dr. Siddhartha Devnath Memorial Oration Award from the Physiological Society of India, and he has very recently, just 1 week ago I think, has received CRS Bronze Medal given by Chirantan Rasayan Sanstha in collaboration with Royal Society of Chemistry of London in a celebration held at Christ University of Bengaluru. I welcome Dr. Kaur Mahapatra to start his deliberations. Before he starts his deliberation, our teachers, rather his colleagues, will give him a ceremonial felicitation.
Thank you, sir. Thank you for your nice, kind, and loving introduction. So 10:30 first hour class is known to maximum of ours, of mine, and maximum students of our student, I guess, nutrition and BMLT, MLT, I can see. From others college also, it's here, I guess, right? Few students came. So before going to start, I must express my sincere gratitude, and it's my honor actually to present today in this college on the auspicious day of our foundation day. So on behalf of the department, I welcome all of you of this session. Respected chairperson, with due your kind permission, I would like to start the presentation. So today it's a nutrition related conference, but my topic actually is related to the infectious disease, which is named the leishmaniasis. So we all know that disease biology actually is two types. One is the communicable disease, one is the non-communicable disease. And both the disease can be prevented through some nutritional aspect as well as their active ingredients. So you can see here that that it's the title actually of this talk is the marine BPMS 22 PPI induced TLR4 dependent macrophage M2 to M1 repolarization augment anti-leishmanial immune responses. So this actually is a BPMS 22 actually is a strain of the Oceanomonas species isolated from a marine source, means the sea source. Okay. So initially that was have some potential activity, then we have proceeded for the others activity against the anti-leishmanial activity. So before I go into my core topic, I must give my tribute to the Sir Upendranath Brahmachari. I think yesterday from his talk, Roku introduced yourself, him. It's a 150th birthday of Sir Upendranath Brahmachari. So he was the first, when he was a student of the MD student, that time he first observed the autopsy sample, spleen autopsy sample, and first he was noticed that something is different within the spleen aspirant. So, okay. Got it. So this is the spleen aspirant which was he observed, and he found something new, and later on his name actually Brahmachari leishmanoid was awarded, means that parasite was known as the leishmanoid. But yesterday he explained the story that actually two scientists, two doctors, parallelly they have identified this parasite, and later Brahmachari actually gave the solution for this parasite. Late 1900s actually, Assam, Guwahati, then northeast region, lots of people are getting died because of this Kalajar. So that time he first discovered that urea stibamine, and it was well acclaimed, and the death rate was become 5% only because of this drug. So this is the first drug which was introduced, and not only that, being the physiology student, so Brahmachari was the founder member of the PSI, Physiological Society of India, and he first established the blood bank also in Kolkata, first blood bank in Asia, and it was first established in Kolkata. So what is leishmaniasis? Leishmaniasis is a parasitic disease. So parasitic disease means it's have the dimorphic life cycle. One is the host, one host is the human, another one is the sandfly. So this is the figure actually for the parasite. It have a nice body and a long tail. So look nice, right? Very good. And this leishmaniasis can be classified into the three different types. One is the visceral leishmaniasis, second is the cutaneous leishmaniasis, and third is the mucocutaneous leishmaniasis. So these names are actually where they are infecting, how they are modifying the pathophysiological changes, all those things. But out of that, visceral leishmaniasis is the fatal one. if it is left untreated. And Indian subcontinent, only the Leishmania donovani is the causative factor. But in other countries like the Brazil, Ethiopia, there you can see the other strains like the Infantum chagasi. But in Indian subcontinent, only the Leishmania donovani is actually causing the disease. So this parasite actually have the two form. One is the promastigote form, another is the amastigote form. Means I told the three different types of the leishmaniasis. Now I'm coming to only the parasitic form. So look, this parasite, that promastigote form has that flagella, means the tail, long tail, whatever you have seen on earlier slides. So that tail actually they infect with the body. When they get inside into the cell, they lose their flagella, and they form into the amastigote form, which is the non-flagellated form. This non-flagellated form within the host cell, they are getting multiplied. So these multiplications actually do not hamper the lifespan of the cell, but it helps the increase of the lifespan of the host cell. In malaria, in contrast to the malaria, what we found, the number of the parasite when it will be growing, then RBCs will be disrupted, then it will be distributed, right? So, but here the cells will be survived for the longer time, and this survival will hijack the immune mechanisms of the host cell, that is the macrophage, and it will induce the severe immune suppression. And you can found the tummy, the blackish skin, tummy, big tummy, means পেট ফুলে যায়, কালো স্পট হয়ে যায় পুরো বডিতে, এই জন্য এর নাম হচ্ছে কি কালা জ্বর. So I'm not going to the epidemiology. The symptoms will be the splenomegaly, enlargement of the spleen, enlargement of the liver. So those are the things apart from that anemia, low platelet count, which are also the common with the others. So there are lots of drugs are available. In the beginning I told about the work of Brahmachari, Professor Brahmachari, but the problem is that that original composition of that drug was lost. Because of that, again the problem persists. Again, and latter which are the companies are now preparing, those are not that much effective what Brahmachari proposed. What Brahmachari proposed, that drug, that composition, that combinations, nothing. It was all lost actually. Later we got lots of others drug. Out of that I should take the name of the amphotericin B, its liposomal formulation ambisome. Oral drug, only one oral drug is there, that is the miltefosine. So others, so these drugs all have the severe side effect. All the drugs, you know that antibiotics, everything have the lots of severe side effect. Low therapeutic index. Low therapeutic index means the cytotoxicity dose and the therapeutic dose, their ratio. It should be above 10, then it will be good. If it will be the below 10, then it will be good, bad. So effective, and not only that, the costing of the treatment also is high. So in that context, actually, we found the Oceanemona species BPMS22 strain, which was initially isolated by one group of my collaborators, Kiran Babu Pulleri, Sastra University. He was actually production engineer. So he isolated several type of strain and offered me, so if I can test something cytotoxicity assay for him. So now actually I'm going to tell a story to the scholars and the project students also who will do the project here. So first time my student, senior scholar in my lab, he told, sir, it is not induce any cytotoxicity. Okay, that's fine, good, give the result to sir, calculate it and give it to sir. He told, no sir, there is another one findings of that. What? No, sir, cells are getting proliferated. Why I am told this? Because simple observations can give you the good lead. Charan came to me and told that, sir, there is a proliferation, huge proliferation. I told, okay, compare with the LPS and others, whatever the mutagens is there in lab. Just compare it and let me know that what is happening over there. So that is the lead to me, actually. We found a lead that this antigen, means the antigen derived from this strain, is a good inducer of the T cell proliferation. So why T cell proliferation? So I think everyone, undergraduate students from the 3rd semester onwards and the PG students, they all know little bit about the immunology, right? Because of their course content. So there we found that there is two things. One is the clonal selection and the clonal proliferation. So clonal proliferation in normally we can see as a proliferation of the cell. Means if the cell, blood cells or anything will be proliferated, definitely that will be the T cell will be the proliferation. So that actually I saw, I did my by hand. Then I thought, okay, chalo, let's do it, something. So because that will be the good lead, and later I gave this work to a project student, MTech project student, Kirtana. So Kirtana worked, tested the crude antigen. Later Harish actually worked, and Harish and Charan did some anti-cancer drug, because we need the fund for the hardcore immunology work. So whatever the Kirtana gave the result, based on that I applied the DBT grant. What I had the only one DBT grant, and in Shastra campus there is only 2 DBT grant that time, me and another one, the Gunjan, 2 Bengali people. So so I'm telling the story, sir. Actually, it will be the interesting to others also, I guess. So 2 grant, and lots of people are telling that Bengalis have the good connections with the DBT, because of that they got the grant. Whatever we hear, when I come back here, I found some myths that, okay, might be the connections with the South Indian people have the good connections with the funding agency. No, that's not the thing actually. So everywhere, whatever it will be, so there will be something, okay, let it be. So then actually we found something good, which we have hypothesized that, okay, it can be worked through the TLRs, and it can induce the macrophage repolarization. So repolarization. So I'm not going to this. So yeah, this slide will be interesting to you actually, I guess. So macrophage phenotypically, so monocytes will be when they will enter into the tissue, they will be converted into the macrophages after their differentiation, and functionally this macrophage can be subdivided into 2 distinct populations according to their activity, right? Suppose after completion of the BMLT or nutrition, what you will do? So either someone will go for the industry, someone will go for the academics. So do you think that who are going to the academics, they cannot come to the industry? Yes, obviously they can do. If the BMLT students, they are doing the work for the pathology, they can do the hematology also, histopathology also. Yeah, if you are working in a certain things, so your expertise will be increased, right? But definitely you can shift to the others. If you are working in an industry and you are getting the 1 crore per year, and someone is offering you that come to the academics and I will give you the 10 crores, you will not do that? Definitely we will do that, right? But our first identity will be whatever we are getting the chance. So similarly, macrophage, whatever they are getting the chance, so they can be either activated in a classically activation, or it will be go to the alternative pathway, that is the M2 polarization. We are giving the name simply way M1 and M2, like the TH1 and TH2. So this TH1, TH2, so if we give the infection, it was well reported that it can convert into the M2 polarization. If we give some treatment or other stimulator like the recombinant IFN gamma LPS treatment, then it can convert to the M1 polarization. So our approach was to, can we do this, that the repolarization from the M2 to M1, because we found that if it will be the converted into the M1, then it can kill the parasite. So that was the concept. So later, why TLR is coming? Look, TLRs have the several, yesterday I think Sangita gave the presentation on the TLRs. So simply during the Leishmania infection, actually the TLR2 dimer will be activated, and it will induce the ERK1/2 and it will induce the immune suppressive cytokines, which will help the parasite growth. So this this 2,2 dimer actually promote the 1,2 dimer also, TLR 1,2 dimer, and it will help the same way. But TLR 9,9 dimer, which is the intracellular dimer, if that can activate it, then it can kill the parasite within the cell. So it will be a little bit difficult for the undergraduate student, but okay, so just know this concept, that's it. Not only that, other TLRs, like, so it can kill the parasite, it can help the parasite survival, but interestingly they have the dual role. So this 2,2 dimer or 1,2 dimer, they are inhibiting the TLR 9,9 dimer, and they are helping for the parasite survival. So others 2 dimer, importantly, TLR 4,4 and the TLR 2,6, they actually can promote the TLR 9,9 dimer, and directly they can activate the P38 MAPK for the killing of the parasite. But similarly, like how they are actually inhibiting that 9,9, so this TLR 2,2 also inhibiting the TLR 2,6 dimer as well as the TLR 4,4 dimer. So there is a very complex things are going to be happen if the TLRs will be activated. So actually from here we have started isolating, purifying the protein, which are it's a downstream actually process. What is downstream? That process, whatever you are going, suppose you are processing some samples, right? So this is the downstream process. So we take the culture, it's a production media we have made up to the 1 liter production media we have made, and from there we have made the sonications, centrifugation, took the precipitate, we run the GFC, chromatography we have done in FPLC. FPLC at first desalting column was used, then anion exchange column was used, then purified fractions was checked in the GFC, that then it was then we have done the affinity chromatogram, and finally we got the purified protein by the ultracentrifugation simply. Then we have done the peptide digestion. Up to this we have done. After that we have sent the sample to the company. So they have tested the fragments, identified the peptide. Then we got the peptide sequences. We modeled them. Then we got the whole protein sequence of that, which one is identified. So the name was the PPI. Why PPI? Because it's a protein protease inhibitor. Protease inhibitor by a protein, so it's a protein protease inhibitor. So initially it was the serine threonine protease inhibitor, and the yield of this we got the, after all the purification steps, we increased the 48-fold of the purification. Means suppose you are getting a activity of the 1 unit after the crude. So if you are purifying that, the others unwanted material will be disrupted, cleared, right? So then the activity will be increased. So like that we have enriched up to the 48-fold of the purifications. Later this one tested of the several experiments, like the promastigote direct killing, then in cytotoxicity, because if we have to treat the or kill the parasite, we have to survive the cells. Otherwise if cell will be die, then parasite cannot be killed by the cell. So that is the immunomodulation actually. Then anti-amastigote assay. Then others M1, M2 markers we have checked, that whether it is converting the M2 to M1 or not. Then signaling pathway we have checked, little bit signaling definitely we have checked. Then we have done the knockdown experiment, which was the targeted TLR4. We have to suppress that or knock down that, then we can confirm. Suppose this is binding with this, so if I disrupt this, then it will not work. So this knockdown experiment actually indicates the same thing. So which is the target, suppress that, inhibit that, then check the activity. If the activity will be disrupted, then definitely we can tell that this is working through this, right? So like that, in vitro and in vivo knockdown experiments we have done. So these are the peak of the GFC. I didn't put the others. This protein have the 35, around 30 KD band we got, and in SDS page we got the 2 subunit, 12 and 15 something we got, and multi-top MSMS analysis actually give the single peak here. Others are the others fragmented because you have done the protopeptide digestion. It's a 30 KD indicated. That protein was identified the mascot software, and it was the histidine amino lyase from the same source. Then that protein was modeled and checked the Ramachandran plot. Their model was good or not in silico analysis. So that protein then tested, it was the protease, right? So protease inhibitor. So I checked that whether it is the inhibiting the protease present in the parasite or not. So there was three important protease. We got the expression we have checked by the RT-PCR, and we found that dipeptyl peptidase can be the target. So it is completely reducing this, the dipeptyl peptidase of the parasite. But later, when we check the IC50, you know that inhibitory concentration of the in promastigote form and the amastigote form. So that gives the another one lead. Okay, so that is the promastigote form, it is 25 microgram per ml. But in promastigote form, it is showing it took the only 0.41 something, means 0.4. How many fold? 50 fold almost. 50 fold. So here is another one interesting thing. So when I got the 50 fold in promastigote to amastigote, and we have purified the 48 fold. So I was assumed that my student have worked with the crude protein, not the purified one. It's a common mentality of the guide. Okay, I have checked personally, I have purified, I have did that experiment again, then it was confirmed, and that time means there was almost why you are not believing us. It's a common to everyone. All the scholars, they don't sometimes believe their guide. Sir, you are not trusting us. No, it's not. It's a confirmation that I have to be, if I am the PI, if I am guiding something, if anyone is executing the concept of ours, then it's my duty to cross check, right? Then that will, what will happen? So by that way, our confidence level will be increased. Our trust on the students will be increased. Okay, so that actually was the interesting thing. So I am shared here. Then other parameters like the inos 2 arginase. Inos 2 will help the kill the parasite. Arginase will help to enhance the parasite growth. So those things we have checked. So those are the marker of the cellular marker which actually help the parasite killing. So now, so these are the image, actually microscopic image, 100x magnification. Look, there is no parasite growth, but it's a huge number of dot. You can see from the paper also. So dot, and this its growth was arrested almost in 1 microgram per ml, means it was almost clear. But believe me that in infectious disease biology, there is a myth of the sterile cure. Means sterile cure is not the possible thing. Now it is, but yeah, definitely some drugs are there which can cure that. But here also, if we go for the percentage, then also we got the 90%, 95%, but 100% we cannot do. So if we compare with the others, then we found that these are the 300 almost microgram per ml, but we got the 25 in the promastigote, and in the amastigote we got the 0.45. Then the others, means all the goods. But yeah, definitely with the standard drug it is nothing, because standard drug look 7.72, but the others, so our is the 25. So huge number of the difference. So now we have to choose that what is going on. So we have designed an experiment where we have to confirm that okay, M1 polarizer, M2 polarizer, infection, and then our treatment, what is going on. So then we found, I will explain only one figure here, remaining will be the similar way. So here 4 set of experiments was there together, control, uninfected, one was untreated, one was PPI treated. Second is the M2, IL-10, IL-4 recombinant protein. If you treat, then it will be converted into the M2 polarized macrophages. And there we gave the PPI. Similar, what happened? that M2 markers are genase 1, it was reduced even after the treatment of the recombinant protein. So that indicates that, okay, look, parasite, their infectivity can be reduced, but recombinant protein, their dose will induce the similar kind of response everywhere, anywhere you can do the experiments. So like that, IFN gamma LPS, which can induce the M1 polarization, but it doesn't induce anything. L Donovani, which is induced the M2 polarizer, and PPI actually significantly reduce the M2 marker. So like that, others marker, CD206, CD68, FIS1, YM1, all those markers has been changed. So we can tell from here that PPI not only the reducing the recombinant cytokines induced M2 polarization, but also it can reduce the infection induced the M2 polarization. Similarly, opposite way, we have checked the M1 polarizer. So you can see here that M1 polarizer is increased. INOS2, IL-1 beta, IL-12, all those things. Some others markers, CD80, CD86, CD40, those names are little bit known, I guess, right? Co-stimulatory markers. So those are actually increased. So those can be considered as an activated marker of this. So those are the cytokines expression. Similarly, M1 cytokines, M2 cytokines, means pro-inflammatory, anti-inflammatory, we have checked by several assays. And finally, some markers has been checked by the Western blotting technique. So Western blotting technique, so many we cannot do because we don't have that much money. So that was the simple thing. So these are also changed accordingly. So from here we can tell that BPMS22 can repolarize the M2 to M1 macrophages, which can kill the parasite. Now coming to the, but what is that? With whom it is binding? How it is actually regulating? So when we check the structure of the protein, isolated protein, it was the glycoprotein, and the well-known review or literature is available that glycoproteins are the inducer of the TLRs or modulator of the TLRs, and that can induce some anti-pro-inflammatory cytokines marker, some cytosolic factor, which can promote the parasite killing. So then we got that, okay, whether it is inducing the TLRs or not. And out of all the TLRs, actually we got the TLR4, only TLR4 is actually inducing in uninfected and the infected macrophages. Some TLRs are also inducing, but it is the only one which is actually inducing the TLR in uninfected as well as the infected. So that's why we have chosen the TLR4. Then we check the docking again, we have checked. Then we have did the only TLR4 Western blot, and we can found that, okay, TLR4 expression was promoted in uninfected and infected. So all this way actually we have confirmed. Later other signaling protein we have checked. Then the time has come to confirm it. How? siRNA can be the in vitro suppressor, and shRNA was the in vivo suppressor. So siRNA we have checked, used to suppress the TLR4 expression, and we found that when we suppress that and we give the treatment, the promotion of the M1 was diminished, and promotion of the M2 is enhanced. So from there we can confirm that TLR4 will be the receptor which is recognizing the PPI and inducing all those things to kill the parasite. So then in vivo experiment again we have done. Here we have initially we have selected the dose dependent studies, all those things we have done earlier, and we have selected the 1 milligram per kg body weight. And here I have played another one things that initially we have gave the 10 different doses, but final experiment we have selected for the only the 3 doses we gave, which is compared with the others reported standard drugs, and we got the good results of it. So here also shRNA actually, where we give the shRNA treatment, there the parasite killing ability of the PPI was minimized. Yeah, so finally these are the some datas. I think we all will be bored about that. So another one interesting experiment we have done to confirm the involvement of the T cell. So here we take the primed T cell, right? Primed T cell. So primed T cell means infected mice. We have collected the T cells. Then we have co-cultured with the syngenic macrophages. Syngenic means same genetically organized macrophages. Then that macrophages were given the infection. Then that D10 means day 10. After giving the infection, day 10 at day 20 we collected, co-cultured. The macrophages were given the infection, treated. Then we found that T cell cytokines marker like the IFN gamma, IL-12 was promoted, but that was also suppressed in presence of the siRNA. So from here also we can tell that TLR4 also involved for the promotion of the T cell proliferation, because that can modulate the antigen presentation, all those things, but I cannot tell because I have not done this kind of experiment, but theoretically we can explain. So finally we have identified what is that which was actually modulating the TLR, which was modulating actually by the PPI, BPMS22 PPI. So after this, this paper actually got published in Frontiers, and first was published in International Immunopharmacology. And I'm taking this privilege to share with you, after getting publications of this paper, DBT officials handler actually retweeted and tweeted that figure of ours, graphical abstract, from their official handler, and so that is the good thing I earned directly. And before going to end, I must acknowledge the when I actually another here actually one story is there, lots of people don't know, students basically. When I came here, the last part of the work was going on at Shastra, and Aditya, I was here, and because of the Honorable Director Sir, he permit me to continue this work here and to give the all support to my student directly for the completion of that work. So, and not only that, I was very much fortunate without sanctioning a single grant to establish the lab here. Because of the utilizing the institutional grant, it was almost not a 35 lakhs initially he sanctioned. So I'm very much thankful to you, sir, and others, my official co-PI, Madam Kuntal sir, moral support, principal sir. Obviously, it is not only that, being the physiologist, Dilip Nondi sir, he was also always admired me and encouraged me mentally, means that promoted other scholars, faculties, Sushmita sir, all helped me to reduce my work, others' work, and complete this work. So later, my funding for this work, DBT, definitely the others funding which actually helped me to build up the concept, that SERB, which was the first grant I received. This is the last grant that sanction letter I didn't receive. That's why I didn't put, sir. ICMR, which was that drug-loaded liposome, which was he is working, Raghu is working. This is my first actually group pictures of my lab or of my research group. So all are established. Barsha is in USA doing the PhD, Joy, Anant in Khan actually is one of the student. He gave the always every almost 15 days cook the biryani, sir. so then and these are the scholars actually. Only the Ramakrishnan actually I asked him to go to the others lab because I am living here. So because of that, and others all are established, well established. And this is my present group. So Raghu, Arijit, these faces are very known to you guys, I guess. And these are the TV people. And this is the lab where it's a frost building and corner. This portion is fully my lab, immunology lab. And thank you for your.
Thank you very much, Dr. Kaur Mahapatra, for your illuminating lecture on a high standard immunological subject using sophisticated technology. I have got some very elementary questions, and I wish you just briefly tell me something.
Sure, sir.
First thing, I wish you could repeat why you tested the efficacy of this PPI in promastigote also, because it is only the amastigote that is
can be
causing the
tested.
disease. Then why did you try in case of promastigote also? First thing. Second thing is amastigotes are living inside macrophages. They have little to do with the skin. As a layman, I think so. Then why is leishmania causing darkening of the skin? And third thing is the amastigotes are living inside macrophages. Then why are these amastigotes not getting digested intracellularly? So I wish you could just...
Yeah, I'm telling, sir, one by one. So at first the promastigote.
fill up my inquisitiveness.
Sure, sir. One is the promastigote. Why promastigote? Because if you are testing the drugs efficacy, so both the form can be tested. Why? Because if it is getting infected, little bit amount can be present extracellularly. It's an early stage, definitely, in early stage. So promastigote, when they actually there are lots of other story also behind to reach after infection to go to the macrophage. There will be the neutrophil. Neutrophil actually and the Langerhans cells, so they are carrying the promastigote to the macrophages. So initially why it is coming? Because of the sandfly bite. So that bite actually releasing the some salivary proteins which are actually increase the melanin secretion, right? Pigmentation, for the blackish pigmentation. Because of that actually full body is getting blackish. So that is the third thing. And another thing you asked for the
Getting intracellularly digested.
that actually the parasite is very much clever and they are escaping the phagolysosomal formation. Phagosome is getting formed, but phagolysosome fusion is actually, that actually, sir, actually there is not that much study on it, and there is no that much hard and core result is there, because there are lots of myths are still there which can be addressed in future.
Thank you. Is there any other question from anyone else? Come on. মাছি কামড়াবার পরে কিছু প্রোমাস্টিগোট বডিতে দু চারটে
থেকেই যায়।
ঢুকেই যায়, তাহলে সেগুলিকেও বাদ দেওয়া যাবে।
বল।
Hello, sir.
Yeah.
Sir, as per a journal issued on November 11, 2012, by a research group headed by Thomas P.C. Dorlo, has been stating that oral miltefosine drug is often referred as a teratogen for some purpose, and it is also considered as a treatment to the leishmaniasis. So I would like to know, we always heard of the word the teratogen, which causes birth disorders. Is there any drug that is having ignorant effect of teratogen but is still effective towards the treatment of leishmaniasis?
Okay, so it's a very good questions actually. So I want to tell you something, that all the drugs efficacy, তুমি তো বোধহয় নিউট্রিশনের স্টুডেন্ট, রাইট?
হ্যাঁ স্যার।
ওকে, তোমাকে তোমার মতন করে একটু একটা কথা বলি, যে এই যে একটা কথা বলি না, যে ওয়ান ডায়েট ইজ নট ফিট ফর অল, তাই তো? মানে একটা খাওয়ার কিন্তু সবার জন্য ফিট হয় না, সেরকমভাবে ড্রাগের এফিকেসিও কিন্তু সবার জন্য ফিট হয় না। একটা ড্রাগ একজনের জন্য, এমনি ধরো আমাদের যে জ্বর হয়, কারুর ক্যালপল 650 খেলে কমে যায়, কারুর হচ্ছে কি তার সঙ্গে একটা সিট্রিজিন খেতে হয়, কারুর জন্য কিন্তু আবার একটা কিছু অ্যান্টিবায়োটিকও খেতে হয়। এইরকমভাবে পুরো ওয়ার্ল্ড ওয়াইড আমি যদি দেখি, তাহলেও কিন্তু আমার ড্রাগের এফিকেসি ভ্যারি করে বাবা, ঠিক আছে? এটা কিন্তু একটা দারুণ জিনিস, মানে দারুণ একটা জিনিস। এবং সেই জন্য যে সবাই বলে যে এত মিলিয়ন মিলিয়ন ডলার খরচ হয়ে কেন নতুন নতুন ড্রাগ আমার তৈরি করার দরকার, ইভেন দেয়ার ইজ নো রেজিস্ট্যান্স অলসো, তাই তো? তো সেই রকমভাবে আমাদের মিল্টেফোর্সিন ইজ দ্য ফার্স্ট ডিসকভার্ড অ্যাজ এন অ্যান্টি ক্যান্সার ড্রাগ। ওকে, সো এই অ্যান্টি ক্যান্সার ড্রাগ যখন ও অ্যাকচুয়ালি একটা ফসফোডিলাইল কোলিন গ্রুপ রয়েছে, ওই গ্রুপটা কি করে, ফিটাল মেমব্রেনকে ক্রস করে, মানে ইন্ট্রাইউটেরিয়ান মেমব্রেনকে ক্রস করে ও কিন্তু তোমার মানে ডেভেলপিং ফিটাসকে নষ্ট করে দেয়, এবং বিকজ অফ দ্যাট ইটস এ টেরাটোজেনিক। সো তার জন্য সেরকম কোনো ড্রাগ বোধহয় নেই। সেই জন্য আমরা প্রেগনেন্ট ওমেনদের, বিশেষ করে ইন্ডিয়ান সাবকন্টিনেন্টে, ইট ইজ রেস্ট্রিক্টেড টু ইউজ। ইভেন মিল্টেফোর্সিন অলসো রেস্ট্রিক্টেড টু ইউজ ইন দ্য ব্রাজিল অলসো। ওকে, সো সেই জন্য বিভিন্ন ড্রাগ, যেমন এখনও তোমার স্টিভোগ্লুকোনেট তোমার কিন্তু ইউথোপিয়াতে ইউজ হয়, কিন্তু ইন্ডিয়াতে চলে না। ইন্ডিয়াতে তুমি যত কেস পাবে সবই কিন্তু এসএসজির হচ্ছে কি স্যাগ রেজিস্ট্যান্স আমরা বলি, ঠিক আছে? তো এইরকম অনেক ব্যাপার রয়েছে। গুড, তুমি পড়াশোনা করেছো বা হয়তো প্রশ্ন করার জন্য পড়েছো। এনিওয়ে, দ্যাটস গুড।
Thank you, sir.
Thank you, Dr. Kaur Mahapatra. Now I request our Honorable Director, Dr. Pradeep
Because
Ghosh,
Dolro is the
to come
well-known
onto the
scientist
stage
group, we know that
kindly
very
and
well.
to hand over a Young Scientist Award to Dr. Kaur Mahapatra. A big round of applause for Dr. Kaur Mahapatra, and the photographers may kindly take some snaps. Now it is time to hand over to Professor Dilip Kumar Nandi to start the valedictory session.
Thank you once again, sirs. Now we will have the award giving ceremony of poster presentation of UG and PG students and oral presentations of the research scholars. We are starting here with the award giving ceremony of the research scholars. I am taking here the names of the position holders one by one.
Now I am requesting Dr. Sudipta Chakraborty, Principal, Midnapore City College, to come to the stage to give the first prize.
First, we can see here the name of two research scholars who have secured the first position in oral presentation. The names are Riya Kaur and Pipika Das. You are requested to come to the stage.
Riya Kaur and Pipika Das. A big round of applause for them. Again, a big round of applause, everybody. Thank you, sir. I'm requesting you to kindly take your seat. Now I'm requesting for the second prize, Dr. Kuntal Ghosh, Vice Principal, Midnapore City College, to come to the stage.
The second position has also been held by two scholars. The names are Priyanka Kaur, Purno Nandi and his group. You are requested to come to the stage.
Come to the stage, please. A big round of applause, everybody. Thank you, sir. Now, for the third position, I would like to request to come to the stage Professor Nirmal Kumar Sarkar. Sir, please come to the stage to give the third prize.
Now the third position has also been secured by two scholars. The names are Raghunath Hajra, Tridip Kumar Das and his team. You are requested to come to the stage.
A big round of applause, everybody. Now it's time for the poster presentation award for the postgraduate students. I would like to request Dr. Pradeep Ghosh, Honorable Director, Medinipur City College, to give the awards. All the three awards will be given by our director, sir. A big clap for sir.
The name of the first position holder of poster presentation of PG student is Madhumita Mahato. You are requested to come to the stage. PG student of Food Science and Nutrition, Midnapore City College, Madhumita Mahato.
Please come to the stage.
Second.
On her behalf, I would like to request Dr. Shravani Pradhan, Coordinator. Madam will be receiving the prize on her behalf. A big round of applause.
The name of second position holder of poster presentation of PG student is Paromita Devnath and her team. PG students of Food Science and Nutrition, Medinipur City College. All of you are requested to come to the stage. Paromita Devnath, Suborna Sahu, Nipa Mahato. Please come to the stage.
His friend will be receiving the award on their behalf from Director Sir. A big round of applause.
The name of the third position holder of the poster presentation of PG student is Mr. Shah Arful Haq. Students of Agriculture, you are requested to come to the stage.
On behalf of the department, I am requesting any faculty member from Agriculture Department to receive the prize. So I am requesting Dr. Suchishmita Roy to receive the prize on his behalf. A big round of applause. Thank you very much, sir. Sir, you can kindly take your seat. Now it's time for the UG Poster Presentation Award. I would like to request Dr. Dilip Kumar Nandi to come to the stage to give the first prize. Sir, please come to the stage.
I am taking the name of the first position holder of poster presentation of UG student. The name is Saptorshi Mondol. You are requested to come to the stage.
Lekha nahi, sir. On his behalf, his friends will be receiving the award. A big round of applause. They are from Bakura Sammilani College. A big round of applause for them. Thank you, sir. Sir, please take your seat. Now I am requesting Dr. Shantanukar Mahapatra to please come to the stage to give the second prize.
The name of the second position holders of poster presentation of UG students are Koel Barik, Debolina Sahu, Sudipta Mondol. All of you are requested to come to the stage.
They are from PK College, Kontai. Please come to the stage to receive the prize.
On behalf of
PK College. Any representative from PK College, Kontai? Any seniors, any representative of PK College from Kontai, please come to the stage. So I would like to request Dr. Shravani Pradhan to come to the stage to receive the prize on their behalf. A big round of applause. Again, a big round of applause, everybody.
the name of the third position holder of poster presentation of UG student is Mohima Dhara, Department of Medical Microbiology, Midnapore City College. You are requested to come to the stage.
And I would like to request Dr. Shravani Pradhan to come to the stage to give the third prize. Any representative who would like to receive the prize, please come to the stage. I would like to request Dr. Suchishmita Roy to receive the prize on her behalf. Madam, please come to the stage. A big round of applause, everybody. Thank you, madam. Please take your seat. Now we have reached the end of our conference. It has been a wonderful journey for us. Ma, it is time to wait for another year. I would like to request Dr. Dilip Kumar Nandi, Professor, Department of Paramedical and Allied Health Sciences, Midnapore City College, and the coordinator of this conference to end our conference with a vote of thanks. A big round of applause.
Now, very good afternoon, everyone. শোনা যাচ্ছে? I am very much, now I am satisfied, as because the auditorium is full of students, even also the guardian and our teachers, that's means the faculties, research scholar. And initially I have very much disappointed. But this is the formal vote of thanks on behalf of the organizing committee. And in this 3 days annual conference, Mcccon 2024 has held in this esteemed college at Midnapore district. At first, I convey my deep sense of gratitude to all of you and specially respect as well as warm thankful to our energetic, dynamic, enthusiastic, honorable director, Dr. Pradeep Ghosh, and honorable chairman, Sri Prabhas Ghosh. Why? For their outstanding contribution regarding expansions of diversified higher education, very important, diversified higher education in backward district of Midnapore under West Bengal in this 21st century. So, big your hands together for their honor, for honor their contribution. Now, our respected chairpersons of this session, Honorable Professor Nirmal Kumar Sarkar, really grateful to him. Actually, she is untireless, the faculty member, always encouraging to the younger students and the researcher and the faculties. My dear colleagues, now colleagues, the faculties, researcher, and the students, for your nice participations of this conference. It my privilege asked to propose the vote of thanks informally. In this occasion, I, on behalf of the Midnapore City College and the entire organizing committee, extend a very heartfelt gratitude to all national and international speakers for gracing your, particularly the international and the national speaker and works, and also the sharing with us your findings and opinions for this 3 days national conference Mcccon 2024. We are really proud and congratulate enough for young, energetic 638 participants, not a matter of jokes, out of which 392 undergraduate students, 225 postgraduate students, 17 research scholars, and 12 off-campus faculties from Vidyasagar University, Sidhu Kanu Birsa University, then Man Sarovar Gobal University MP, then Bajkul Mahavidyalaya, Belda College, Bankura Sammiloni, Prabhat Chandra College, Raja Narendralal Khan Women's College Autonomous, Midnapur College Autonomous, AKBC Mahavidyalaya, then Nistarini College Purulia, then Amdohi High School, Borjora High School, and also from CMOH office in Purulia. This year, organizing committee has introduced Young Scientist Award, very interestingly, for in-campus and off-campus scientists, mainly offered to Dr. Shantanu Kaur Mahapatra. Already you learned many things from him, the associate professor as well as the head of the department, Midnapore City College. In another faculty is Dr. Kaushik Das, assistant professor and HOD, Belda College. So we hands together for encourage them and also encourage the innovative idea by our Honorable Director, Dr. Pradip Ghosh and his team. Big hands together. May I so many congratulations, one students in the PG students only. The 3 days continuous interact in the seminar. I am very much proud for this. The Deep Chaturaj, the NUD student, really, you are not only a selfie selfie taken not in the students or not in the students as a wastage your time in your mobile, hence your mobile. So I wish you may go in Broadway. Thank you, everyone. I would like to acknowledge all the sponsors, including Department of Biotechnology, DBT, CSIR, Science and Engineering Research Board, SERB, for organizing this event successfully. I would like to express our appreciations to our guests who have taken time out of their busy schedules to grace the occasion with their presence. And your presence has added an extra spark to this event and made it even more special. Professor Anil Kumar Onal from Asian Institute of Technology, Thailand, a true scientist using their knowledge in the developing of the nation and which would be clearly understood for his lecture. Obviously, you had enjoyed his lecture. I would also thankful to our another national scientist, Professor Dr. Madhumita Barua, the Professor, Department of Agriculture and Biotechnology from Assam Agriculture University, a specialist in agriculture biotechnology. Her talk involved how Northeast Indian agriculture fermented products has developed it from time to time with their functional food properties helps in the human health. Really, we very much grateful to her. Another scientist from the ICMR, namely Dr. Dept Bhattacharya in the Regional Research Institute of ICMR in Odisha, Government of India in Odisha. Thanks to Lord the giving us the precious time in our busy schedule, in your busy schedule. I sincerely thank the guest of honor and the speaker for taking time. Really, we are very much grateful to him, Dr. Dev Prasad Chattopadhyay, former director, ICMR, National Institute of Traditional Medicine. Really, his speech is very much informative and applied. The someone is theoretical, but someone is the applied. And he has a lot of experience regarding application and his contribution to the society. So we are very much grateful to him. And even also we are very much proud, particularly myself is the proud, for Professor Keshav Chandra Mandal, the head and the professor in Vidyasagar University, Department of Microbiology. He nicely presented for the functional food applications as a generations of a entrepreneurship itself in the food processing system. Very good. And sharing in this seminar. We are also thankful and also the proud, particularly for the Professor Mandal. Again, I take this opportunity to extend our most sincere thanks to the Dr. Pradip Ghosh, founder, director of Midnapore City College, for his constant support and innovative way of thinking to organizing such annual conference each and every year, last 4 years, I think, in also the continue in the near future. And may I request also, in the next time, you may introduce the lifetime achievement for the scientists or the faculties to honor them in surrounding in Midnapore or the other part of the country. I would like to thank Dr. Sudipta Chakraborty, principal of this Midnapore City College, and Dr. Kuntal Ghosh, Vice Principal of the Midnapore College, and Assistant Professors of the Department of the Microbiology, and all staffs of the college. Really, so many staffs, actually, those are associates with the education. And need not mention the particular name. They are also helped at a lot. But the Abhishek, the Sumon, Madhumita, and like that are more. I would like the thanks and the proud of the organizing secretary. I am very much proud. The continuous initiative taken by Dr. Shravani Pradhan, Assistant Professor, and also joint convener, Dr. Suchismita Rai, Dr. Maloy Patsa, Mrs. Manisha Fuzdar, and others faculties, researchers of the Midnapore City College, Department of the Paramedical, and also the Allied Health Sciences. Words are not enough to thank their constant guidance and the support to shape as well as the success, the such conference. We are very much thankful to Dr. Urpita Raj, even also Mr. Abhishek Chakraborty would be a doctorate, and also the Sormila Rai, Dr. Sormila Rai, Assistant Professors of Department of English, for their pleasant, really pleasant anchoring and coordination and the cooperation in this conference. I would like to thank our beloved, all the organizing members for their constant help and also the guidance, and radiated a source of energy within us. We will ever again grateful to you. We thank you for being with us these days. And thanks all staffs of the Midnapore College, City College. I would like to thank the event organizer, planners, developers for the organizing this event, such a short notice, and also making it such a beautiful and also the memorable event. We have been fortunate enough to be backed by a team of very motivated and dedicated research scholars and the students, committee and the volunteers. And last not the least, my special thanks, really special thanks for cultural team, and headed and the leading by Titli, Panchali, Ananda, and Ria, and perform nicely as per comments of the outside speakers and the resource person. So very much, and wish you best for your in future. Thanks to all for making this event successfully. Have a wonderful staying at Midnapore City College with a green campus and nicely enjoy. Thanks to all.
নমস্কার, আমরা বাঁকুড়া সম্মিলনী কলেজের শিক্ষার্থী। তো প্রথমেই আমি ধন্যবাদ জানাই এই কলেজ, মেদিনীপুর সিটি কলেজের অথরাইজেশনকে, যে এত সুন্দর একটা সেমিনার আয়োজন করার জন্য। এই 3 দিনে যা যা কিছু হলো, যা যা প্রত্যেকটা স্যার প্রেজেন্টেশন দিলেন, সেখান থেকে অনেক কিছুই আমরা শিখলাম। এবং আমরা আগের বছরও এই কলেজে এসেছিলাম এবং এবছর আমরা মোট 13 জন আসতে পেরেছি, তিনটে পোস্টার প্রেজেন্টেশন নিয়ে। তার মধ্যে একটা পোস্টার খুব প্রশংসিত হয়েছে, এই জন্য আমরা খুব গর্বিত বোধ করছি। এবং আজ এই কলেজের ফাউন্ডেশন ডে, তো আগের বছর এসেছিলাম এবং এই বছরে এসেছি, তো এই বছরে দেখছি 2-3টে ডিপার্টমেন্ট এক্সট্রা হয়েছে, তো এই কলেজের উন্নতি হচ্ছে এবং উত্তরোত্তর উন্নতি হোক, এগিয়ে যাক, এই কামনাই আমরা করি। এবং আমরা যে পোস্টার প্রেজেন্টেশন করছি বা আমাদের যে ল্যাবরেটরি যা সমস্ত কাজ হচ্ছে, সমস্ত কিছুই আমরা আমাদের রেসপেক্টেড ডক্টর সুরজিৎ মজুমদার স্যারের কাছ থেকে শিখি। তো স্যারের অথরাইজেশনেই আমরা সমস্ত কাজ করি এবং আমাদের কাজের মধ্যে যে স্ট্যাটিস্টিক্যাল ব্যাপারটা আছে, যে স্ট্যাটিস্টিক্যাল সমস্ত কিছু হয়, সমস্ত কিছু আমাদের সিনিয়র দাদা শুভজিৎ মাহান্তি দা খুব ভালোভাবে রপ্ত করেছেন এবং খুব ভালোভাবে শেখান এবং পুরো ব্যাপারটা দেখে। তো তাকেও ধন্যবাদ আমাদের এই কাজটা দেখার জন্য এবং খুব অনেক ধন্যবাদ আমাদের কাজটাকে অ্যাপ্রিসিয়েট করার জন্য। আমরা যে কাজটি করেছি, তো এই যে এবং অ্যাপ্রিসিয়েশনটা পেয়েছি, এই অ্যাপ্রিসিয়েশন থেকে আমরা ভবিষ্যতে অনেক এগিয়ে যাওয়ার চেষ্টা করব এবং নিজেকে ভালোভাবে প্রতিষ্ঠিত করার চেষ্টা করব। ধন্যবাদ।
We have reached the end of our conference. Right now we have with us Mrs. Anindita Ghosh, Vice President, Midnapore City College. Madam is here with us. So a big round of applause for Madam, as she will be with us the entire day for the College Foundation Day ceremony. Now it's time to shift our attention from the seminar hall to the college ground. Under the leadership of Dr. Pradeep Ghosh, our honorable, respected and loving director, sir, we have reached this far. A big round of applause for sir. Now, I am requesting everybody to take part in the ceremony to the college ground to enjoy our College Foundation Day ceremony. Thank you, everybody.